Hello and welcome to Peter Hill Explains, where I discuss things scientific and just in general get practice teaching. If I don't have a class to teach you, I'm going to actually focus on developing podcasting skills and video skills. So sorry to you for being my guinea pigs. This time I'm going to address something called a neuron, which is the basic cell inside your brain and inside long tubes, say, your nerves. So your nerves will go from your spinal cord up. But it won't be a single neuron. It'll be a series, a whole bundle of neurons, a bit like a data cable. If you open up, there's many different wires that's connected together. And what I'm going to do is give a series of activities, scaffold activities, from the very simple to the more advanced stuff. Now, typically, as I've read into the more advanced stuff, I've said, hang on, that's not correct, or that's slightly different from what I've learnt about. So I suppose in general, students learn that the brain is just full of neurons and nothing else. And there's a few things to do with that just how neurons work, and they really struggle to understand. They learn pretty much the central nervous system, which is your brain, your spinal cord, and your peripheral nervous system is everything else, which goes out to the actual functioning bits. The central nervous system is more the management structure, and then the brain is really, you could really consider the spinal cord just as an extension of the brain, because actually brain parts actually start to appear as you reach the top of the spinal cord. You get the brain stem and things like that. We'll talk about the cerebellum and the cerebral cortex. They're different parts of the brain. In fact, when you look at it, I'd say, well, we've got at least 2 or 3 brain parts, and what actually has happened during evolution is that we've called out, we've had a spinal column, and then we've added onto that, so we've added more sophisticated brains. The most sophisticated brain is the frontal lobe. That's the most advanced part, and that's become the most recent. And of course, that's added on, sort of like a shopping centre or a town, and where rather than knocking down anything old, you've actually added more and more on to it. And I should also say the dual purpose of this is to be able to give this as a podcast, but also as a video. So the podcast will mean that I will tend to describe things in fairly big detail and explain where I'm going to, whereas a video you can actually hop around by just showing things on the screen. Now, obviously, if you listen to a video, it's very hard to pick out what's going on, but if you have a podcast which of a video seems rather dull and boring, so I hope I'm not too dull and boring. So the most basic thing you want to do, you've got students in, you discuss perhaps what neurons are and things like that, but to actually bring or let them make a model of a neuron, and I've actually done it here. So I'm leaning over and I'm picking up a silver foil model, or aluminium foil model of a neuron, and it's not an artistic wonder. I've just got about 2 feet of aluminium foil and scrunched it loosely up, and then with scissors cut up the various parts. And so you'll see something which looks here a bit like a neuron. It's not sophisticated, but it's there in front of you, and you have to actually go through the skills of identifying the various parts. So what I'm going to do is, firstly, I'm going to sneeze. Achoo! That is some irritation that has affected my neurons and tickled the neurons, and it's gone through and created a set of reactions, and it's eventually sent off to my muscles a response. So that tickling is very much what a neuron does. Neurons are basically sneezing cells. That is, they have several components. So I'm pointing over here to these little loose bits of aluminium foil as best I can do at the very end. So I've just left the last end of the aluminium foil as a cone, then I've cut it up and squished it up, and those are called the hairs of the cell, or the dendrites. dendrites is Latin for hair. These are different cells, hairs, and as these get puffed with neurotransmitters, the actual chemicals from the synapses or other neurons, the electrical pulse builds up on them, and depending on the actual shape and surface of it here, the capacitance of it, it will reach the cell body. So I'm pointing to a clump of silver foil. It's not so big, I've not squished it down, so I've actually put my finger in so I can build it up, and that is the cell body. This is the configuration for a transmission neuron. That is, things come in this way, go through and out through the synapse. So this transmits a nerve pulse through it. Now in the cell body you've got the machinery which keeps the cell alive. There's actually no sugar storage cells in a nerve cell, so a nerve cell has to be mothered all the entire time. It has to be looked after, and we'll discuss that in a bit further detail. It's got the nucleus and other components inside there. But the most important thing here is that this body makes a decision. So it determines the amount and quantity of stimulation before it decides to fire. So it fires a nerve pulse down a long slender thing. So I'm pointing to some silver foil which I've squished down into a long pipe. In this long pipe it will decide to send it. So what a nerve cell does, is it calculates the amount of stimulation. As the stimulation reaches a certain threshold, it will fire, and the amount of threshold can be changed very quickly, it can be programmed very quickly, instantly, by actually just changing the shape of these dendrites, the speed at which the pulse goes down the dendrites. So we've got the dendrites, the cell body, and coming along here we've got the something called the axon. The axon is the neuron fiber, the nerve fiber as it's called. So this is the thing which actually transports information, and you can have, I think in your body there's one several meters long, and in your brain they travel a short distance or a long complicated distance between the two halves of the brain. And that's very interesting that besides having multiple brains, the sort of ancient brains into more modern brains, the actual modern brain is divided into two halves. So you've actually got two halves, two personalities working together. In fact, if I was to cut the nerve fibers between two parts of the brain, they have found that people maintain two personalities at the same time. A affective personality, which sort of explains socially what's going on, and an analytic one. We'll have to get into that a bit more. So the nerve fiber goes along here. It's like a series of domino effect of batteries. It looks like a pulse, but it's actually a series of chemical reactions which go out here to the synapses. Synapses have little feet on them. The nerve goes down here, splits out, and all of these spread little feet on them, produce the synapses. So the little bulbs with chemicals can be pushed through, so the chemicals build up. When the pulse hits them, they actually fire through. So imagine this is a bit like a pepper shaker. So if I want to sprinkle some neurotransmitters, I brush this section up here, tickle this section, it goes achoo, and it sends it all the way down here to these pulses around here, and this fires off, and it fires off doing neurotransmitters. Of course, the next one's here, and it's going to have several nerves all feeding it at the same. So I think this is a good model. In fact, being out of silver foil, you can actually get people to connect the nerves together and light up a light bulb at the other end. Maybe a bit confusing. What I've also done here is actually put this, which is the instillation. So this is the raw axon, and you have to wrap instillation, and it's called myelin. And this is an instillation, a sort of fatty film which actually wraps around and around and around. And there are certain diseases in which this falls away, and you get a brain short circuit, or you can get just material building up, destroying the ability to look after this. And what I want to do is actually, that's the easy part and quite a fun activity just to introduce it, where we've introduced the central nervous system, the peripheral nervous system, and the basics idea of what a neuron is with a basic model. And it's here when I was looking at the myelin that I realized that there's a lot more, and I heard some science show stories and saying that something like only 10% of a brain is actually made out of these guys. They're the I suppose, if you like, the exciting aspects of nervous systems. They are the nerve, it's the nerve system, but there's a lot of support stuff. So if you can imagine, the nerves are the runners in the race, and there's a whole lot of other backup teams, people manning the drink stations, a whole lot of marshals checking everything's all right. There's police keeping the crowd back, keeping bad guys away. There's a whole heap of things. There's a whole heap of other cells which exist. And the term for it, I hope I can, glia is the actual term, G-L-I-A. It comes from the Latin of glue. And glue comes up a lot of times in biology. Gluten means the glue protein. Amazing. Glia is when they first came it up, they said this is the glue, and this is actually correct. Now, I've got the myelin here, and it was a bit distressing that I found out that people used to draw this thing with the dendrites, I'm pointing to the various parts here, the bulge here, which is the cell body, which does the processing, the axon, and the synapse is going all different directions, and then put the myelin sheath right over the top here, and they actually put cells here. But the interesting thing here is that the myelin sheath in your long body actually is in fact little cells sitting there because they're not interconnected. But in the brain, there'll be a cell called, just trying to actually think, something dendrite or something like that. But it's actually a cell which sits out here, and it's got little fibers out, and the end of the fiber is the wrap of the myelin. So there's a cell out here inside the brain, a cell out here, a different cell, I can't pronounce it, I'm sorry, but it's another cell out here, and so for one cell here, it will send fibers out and do a little bit of insulation on half a dozen cells. And obviously this will hold the brain, the things in place. It's half holding in place. So there are two things: the actual cell, where the actual cell walls there, and it does the local thing, is for your peripheral nervous system going down you, and that's called a Schwann cell. And I sort of remember reading something about it. I can't quite remember at the moment. So that does insulation, but there's also other things. What I'm doing at the moment is something called a star cell, astrocyte, or something like that. It's a star cell because it's star shaped. And what it does is it sends up a support here and a support around here. And what it also does is it goes down and actually hermetically insulates the entire brain by actually putting a coating around every capillary, bringing oxygen to the brain. So if somehow these astrocyte cells so these never see blood. Blood doesn't get into the brain. So it's a blood-brain barrier. There's no blood in the brain. This is pretty much, and this is a cell that needs oxygen. And so there must be some sort of oxygen transport from these star cells, astrocytes or something like that. And because I've only read it, I've never heard it. So the astrocytes here and they hold a whole series of cells together, and they go and they clamp around the capillaries and actually seal them off, so nothing can get out of the capillaries. So your brain is a pure thing. So they're the two things which actually support the cells. So there's also, similar to astrocytes, cells in the central nervous system which feed and look after the long haul cells, like the Telstra cells. Oh, I call them Telstra cells. The NBM, long fibers, which are long distance, but when you get into the junction box, everything goes around. So you've got the support cells, and you've got the installation cells. And inside the brain, one installation cell will go off and do 30% of three neurons around it. Very interesting thing. It's, what is it? It's not good for school because it's complex, but then again, it's the truth. So you've got this balanced thing. Do you actually simplify things so the kids can go, yep, and tick off? And then it's more complex. How do you do it? Perhaps talk it through in a video like this. But there's a few other cells. There's specialized brain immune cells. How do they work? Not quite sure. But they're pretty much like white cells. And they're microglia. I haven't got the pronunciation of glia correct. But these are little monster shells, and they walk around your brain. They're not fixed at a point of the cell, because what's the point of having an immune cell which is fixed? You work out to fix one infection, the infection goes to the other part of the brain. These cells walk around the inside of your brain. And if there's any damage, which from injury or build up of plaque or things like that, they'll walk around and clean it up. And this is, I suppose, an important thing. It's believed that when you sleep, these cells wake up and walk around your brain. Or at least I've read a few articles proposing that. I'm not a neuroscientist. The thing is to actually start asking it. And then there's other cells. So your brain is in a blood-free bag. There's got veins inside it. And when you shoot someone, there's blood goes everywhere. But the brain in operation is actually sealed the installation, and which is the these central ones with the myelin going in. There's cells which support the cells, but also seal off the circulation of it. And finally, there's another type of cell called the ependymal. I can't pronounce it again. Ependymal cell, and that's if it's not blood, it needs other things, another fluid. You've got the spinal fluid. So when it's going to the blood-brain barrier goes around the brain and down the spinal cord as well, and your spinal fluid is inside, and the fluid has to circulate. It moves rubbish around, it cleans it all. It's like a mini body system inside, and these cells have little hairs, and they brush the fluid in your head. And your brain has these cavities, liquid cavities around the hippocampus, around there. There's liquid cavities. And I've basically held a brain, which has been a fantastic thing to actually. It's not a fresh brain. It was in formaldehyde held for a while, but actually holding it, seeing all the cavities and complexity around it. So there's the neuron transmission cell with the dendrites. I'm pointing to the little hairs at the end, the body, which actually does the computer part of the cell. So this is the input, computation, output, transmission line, and transmitter. So it transmits, and the myelin coating. And I've done this as a continuous coat. Like I've got this coat here. It's not there's little hands, little hands going out. So if you can imagine, I'm one cell, and I'll grab one like this, and there'll be another hand which will grab up here. Another cell around will grab him in like that. So there's a lot of cells, and of course the health of these cells, it's vital. It's almost like another another being living within you, inside your brain. It's very fascinating. There's a lot of detail there, and I've been thinking about it sort of like you wouldn't teach it in a school, but surely we should structure things more such that depending on, there's so much information there and so much beauty and complexity there that you go through, that there's so much knowledge there, that rather than teaching a sort of a single little area, making sure that we can test it, having a much larger area which we can progress through depending where the students get to. They might be interested in neurons or the glia cells, the host of other fascinating things which if you were to ask curious questions like, how does the insulation get there? How does it support it? How does it clean up rubbish? How does it maintain the blood-brain barrier? If you had inquiry learning, you would have to break out of this small region that we do teach at. And there seems to be two answers to that. One is to clamp down on discovery learning or expanded learning, or to actually think of a new way to go ahead with new people and a new idea of how to teach it. That's an episode of Peter Hill Explains, purely off the top of my head. Just having reading, I decided that I should teach on this, and this will reach out to people who are interested in neurons and teaching like this. But in the meantime, so I'm doing this as practice, but in the meantime, do what I do, is keep on doing science, building models like this, reading research, keep on thinking science, trying to actually place it together, and then finally experiment as a scientist, working out better ways to teach and communicate with people, because that's all you can do. Thanks for listening.